over to Jake.
Thank you very much. Thanks for that introduction. I will be sort of practicing what I preach with that quote and showing you apparently unproductive work that has been the beginning of my PhD, which is, as Beck said, in the Grange lab at the Rosalind Franklin Institute. And this work is moving towards applying traditional structural biological techniques, such as cryo-ET, in more complex systems, such as mammalian brain. So there's a range of model systems available to perform structural biology, from the more in vitro side on the left, which is purified proteins for classical techniques such as X-ray diffraction and single particle electron microscopy, through cell culture methods, so 2D cells grown on grids, or cells grown in 2D on grids, and then primary cells, and then you get more complex systems such as 3D cultures and organoids, and then finally, the most complex yet most biologically relevant system is studying molecules in the tissues themselves. But with this increasing biological relevance, you sort of get a direct increase with experimental complexity, in inverted commas, because I'm fully aware that all of these techniques have their own difficulties associated. So the sort of ethos that we've been working towards is how can we make experiments at this end of the biologically relevant spectrum more complex and more sort of approachable to the field. So yeah, so the overarching aim is to achieve routine structural investigation of tissues using cryo-ET, which for those who aren't in the field directly, it looks a little something like this. So you have a cell grown on a grid normally for intracellular cryo-ET. You prepare your lamellae from FIB milling, so an electron-transparent window into the cell, and then you acquire your tilt series, and then you back project them and reconstruct a volume. Things get a little bit more complicated when you start studying multicellular systems. So here on the left-hand side, you have a block of tissues, and you basically have to use a system called cryo-lift-out to extract a smaller region, as you can see in this video in the middle here. So you use the FIB and a manipulator to come in and basically pull out a chunk of tissue, which you then will go on and thin to electron transparency. So you extract the region. This unlocks this new, more complex multicellular samples, and then you serially deposit them for spatially related samples in Z. So your lamella will come from a certain area of tissue, and it will all sort of be related spatially. So in the field recently, there's been a sort of uptake in both the hardware and especially cryo-lift-out in generating structural information from complex systems such as the Elegans embryos, which was sort of the flagship paper that kickstarted all of this, through extracellular matrices, mouse tissues, and egg chambers. So the use of cryo-lift-out is sort of common in all of these studies, and this is opening the possibility to structurally characterize increasingly complex multicellular organisms in previously unattainable detail. So brain tissues are complicated. So we take a whole brain. Oh, these images haven't come out very nicely, but you can imagine you take a slice and a biopsy. Within the biopsies, you have synapses, and then within each of these synapses in the brain, you have sort of this soup of protein that is all vital for its function. And then even within sort of these synapses, you can like locate down to like a synaptic vesicle. And again, the molecular detail here is really quite intense and quite deep. So can we begin to sort of unlock this molecular detail instead of in single cells? Can we take brain tissue and unlock this molecular detail? So this is just from mouse to microscope, the workflow that we've been establishing at the Franklin so far. So at the beginning of this workflow, sample preparation is key. And before you can start performing these experiments with the cryo-lift-out, you have to make sure your sample is preserved in its native hydrated state properly. So, as I say, we take a sagittal section of brain and we take 100 micron slices in the vibratome. And then from here, our primary method is high pressure freezing for vitrification. So we take the biopsy and place it in these copper gold high pressure freezer carriers and then subject it to liquid nitrogen at 2000 bar in order to increase the cooling rate and ensure more thorough vitrification. And the discussion panel earlier, Maud made the comment that we should show sort of the failure rate or the success rate. And I don't really have time to go in detail, but this is work primarily done by Callie Glynn, who's in the audience as well, on how many times we had to optimize this freezing protocol to get like bona fide vitreous brain samples. So this was a good couple of years of work just to get the tissue samples in a position where they can be analyzed by cryo-ET. So that's just a snippet of what we had to go through to get it where it is today. But eventually we did get it vitreous and we got it reproducibly vitreous. So after that, we had to find our region of interest. So you take your highly pixelated schematic of your hippocampus biopsy and you put it in the SEM after high pressure freezing, and you basically have this big sheet of ice, which is featureless. So the question is, how do you find your region of interest? And it's effectively like finding a needle in a haystack. So our approach has been focused on taking the high pressure frozen sample and milling these sort of fiducials into the surface. And then by using the integrated light microscope, you can correlate these SEM images with the fluorescence. And this is just a slice of hippocampus, which has been stained with Hirsch, which can identify the nuclei, which because the hippocampus is such a well-studied region of the brain, it has a canonical structure, and the cell layer lines, you can sort of see, my point is not working, but you can plot the cell body layers, and then from there you can identify regions such as the CA1, CA2, CA3, and dentate gyrus. So by using a small chemical dye, we've sort of orientated ourselves to find regions within the brain for lift out. So there's been a couple of strands of sort of optimization going into the lift out protocol for tissues, and one of them is this, what we're going to call a perpendicular lift out. So here's a schematic of the high pressure frozen sample. Top is the top down view, and then on the bottom, if you look through the side, you can see the tissue on the bottom, and the cells are oriented sort of this way from the slicing. So if you took, like you see that the box there, you took a sort of ice core from the sample, you get your cells in a block of tissue, as you can see here, with them orientated like parallel to the chunk, perpendicular to the chunk, sorry. And then you can take serial sections across this block of tissue. And here is just this in practice. So you have a fluorescence, the same fluorescent sample as last time with the cell layers stained. And you can identify the CA1 stratopyramidal and the stratum radiatum. And you can find a region that you're interested in and bring in your needle and take out a piece of brain tissue like this, which is then serially deposited onto a grid. So you bring your block of tissue in, you trim a small bit off, you weld it on with redeposition welding, move it over and repeat until your block of tissue is gone. So you have all of these lamella in a sequential fashion that you can then spin to electron transparency. And here is just a representative tomogram that we have of a synapse in the stratum radiatum. So you can see the left, the blue presynapse with these synaptic vesicles, and then the magneta cleft in the middle, and the purple or blue as it looks on the screen is the postsynapse. And on the left-hand side is just a still, and you can sort of see the level of molecular detail that we can pull out of these images. So you have synaptic proteins within the cleft and microtubules with interluminal bodies, and synaptic vesicles with proteinaceous density on them. So a really great level of detail that you can get from these images. So that's just an example of one of the approaches for the cryo lift out, but another approach is if you take your same slice of high pressure frozen material, but instead of from a top-down perspective, your region of interest goes parallel to the sample plane. So you'll need to play with the geometries a little bit. So if you imagine you take your cells like this, and then eventually make your sections like this. So it involves a 90 degrees rotation, but you can't actually do that with the hardware. So you've got to basically lift it out a certain way, which I'll try and explain now. But here is how schematically that looks, where you'll have the layers of tissue going this way, and then you take your serial sections, which are the gray blocks. So you get a different perspective of the tissue organization. So here's just a slight technical side note of how I managed to extract tissues for lift out like this. So you mill basically extremely large trenches on either side of your region of interest. As you can see, that finger down at the bottom is the region identified. Then you move the stage 90 degrees to the FIB and perform these undercuts to free it from the top of the surface. And then you basically bring your needle in, as you can see on the bottom, and pick out this large lift out from the sample. And it's important to sort of follow on from Pavel's talk, where he used plasma FIB for his imaging and serial FIB SEM. We are also using the plasma FIB, the Helios Hydra, and I think without the use of the xenon plasma in particular to sort of speed up the excavation of this material, all of this method wouldn't be possible. So plasma FIB, if a take home is, plasma FIB is essential for performing structural biology in thick specimen. So I showed how you would get the sample, and the idea was, can we sample across a whole, several layers of tissue with a single lift out? So again, we have this fluorescence image where you span the different regions of brain. You isolate your region, and then you come and pick up this 370 micron long cryo section. Side note, this thing is ridiculously big. You can see it from the chamber cam in the microscope. This is this enormous lift out compared to a regular lift out, just for scale. So yeah, it's big. And then from here, you do the serial deposition, apply it to the grids, which you see by the welding, and you end up with these huge numbers of serially deposited material from a single lift out. And you thin it with a combination of xenon and argon, and then you take it to the TEM, and you can see this is a medium magnification image, and you can start picking out features for targeting, for example, dendrites. So from this perspective, the reason why we did it is to get these cross sections of the dendrite network in cryogenic specimens. So, and because they are all sequential, this is technically volume EM. So this is for the volume EM aficionados amongst us. And then just a quick comparison, we have sort of previous attempts at looking at these dendrite networks in the resin embedded world, where you can see these rings with the microtubules inside, and then through high pressure frozen, yet with a microtome, you can also make out these microtubules, but the resolution isn't super great. But this is our sort of attempt at this, and in my opinion, you unlock an awful lot more of the molecular detail of the cytoskeleton network and individual proteins inside these dendrites, which are amenable for further processing, such as subvolume averaging, to get high resolution structural information out of it. And then finally is just some examples of that high resolution information. You can look at the microtubules and count the different number of protofilaments. There's incomplete microtubules, so it's evidence of dynamic processes. There's interluminal bodies with protein linkers inside the microtubules. There's linked microtubules and linked with a protein bridge. There's interactions between the microtubules and the endoplasmic reticulum with, again, protein linkers. And then these large networks of actin bundles that go through the volume. So unprecedented molecular detail. So yeah, to summary, we've developed this workflow to do structural biology inside tissues that's reproducible and high throughput. We can use CLEM to target regions of interest without an endogenous fluorophore, which is important for human tissue samples or biopsies. We achieve decent contrast without any toxic heavy metals, so it's just ice, which is nice. And again, these connected spatially images from the serial lift out is acquiring cellular cryo-ET data over a volume. And if you can vitrify it, in my opinion, you can image it. So the main bottleneck is getting your sample vitreous, and then hopefully you can apply these methods and get nice images like this. So big thanks go to mainly Michael Grange, who's my supervisor, and Callie Glynn with the big red arrow, because she missed the photo, who is the co-author on this preprint that just came out last week. If you are interested, give it a scan. And thanks to the rest of the group and the folks at the RFI. And also we have Victoria Garcia Jenner, who has a poster at the back as well, if you want to talk more cryo-ET developments. And with that, I'll say thank you and take any questions.
Thanks very much, Jake. Any questions from the audience?
Do you prefix your brain before cutting on vibrotome?
Do you prefix?
Yeah.
No, it's just straight fresh from dissection and then into hemispheres, straight on the chuck.
Kill a mouse and take the brain and slice it and stain it with Hearst.
Yeah.
And then put it in.
High pressure
How long does
freezer.
it take?
We normally do several sections, so it takes a little bit longer, like an hour, maybe 2 hours. But in theory, you could slice. One slice takes about 30 seconds and then run downstairs and freeze it within minutes. But we tend to do serial sectioning, which takes a little bit longer, and then freeze them all at once. But there's no reason why you couldn't take the brain, immediately freeze it within minutes.
Do you have any signs of autolysis? Does it take the brain, cut down the begins to die?
So we have been really diligent in preserving as well as we can these tissue slices, and we take our inspiration from like the electrophysiology field. So they often take sections of live brain and do patch clamping for hours, maybe 6 or 7 hours. So it's all about keeping them in oxygenated buffer that's pH controlled at the right temperature. So that's all done before the freezing step.
Very nice talk. I was just curious, when you do the serial slicing with a xenon, how thin was each serial slice?
So for us, we tend to do 4 to 5 microns. The plasma beam itself has quite a large beam profile, so you're always going to have some missing information between, and there's always a limit on how thin you can actually do these sections without sort of playing with the hardware side of things because of just the nature of the beam. And also, if you do a lot, it depends what your question is. If you want to cover a number of layers of tissue, if you do really thin slices, you're going to be there forever actually acquiring the TEM data. So you can sample sort of thicker to get over a greater range of tissue areas, or you can do a small region of thinner samples, maybe 1 micron if your beam is well behaved. So it's just a trade-off between the two.
Great. So if there aren't any more burning, well, Pavel's got one.
Hi, thank you for presentation. I'm wondering, on your vertical lift out, the first lift out, how do you protect the leading edge of the block? So how you deposit the protective layer?
You mean for making lamella afterwards?
Yeah.
So basically, instead of going through the front, imagine your block, we take a sort of shallower angle through the top of the surface that's gissed afterwards. So you're not making it through the curtained front, you just tilt it, make a bit of a deeper lamella, but you have a smoother surface to make that on. Because we tried making it through the front and they just fall apart. So.
Okay, great. Let's thank Jake again for a fantastic talk.